Today it's on to day 3 of the lab. That's where you're going to view the genes that you've copied through PCR. The way we're going to do this is through gel electrophoresis. Gel electrophoresis is a really visual process, so the best way to show it to you is through other videos. So you're going to watch explanatory videos on gel electrophoresis, but specific to the lab, we are going to take our 6 PCR tubes and we're going to put 10 microliters of orange loading dye into each one. Sometimes loading dyes are blue, sometimes they're orange. The important thing is it makes the sample visible in the wells of the gel. Then we're going to add one more tube, and that's a PCR molecular weight ruler, and that's going to show us fragment lengths or a rough idea of fragment lengths. We'll take those 7 samples and put the gel into the buffer and into the gel chamber as shown in the other videos. And then you're going to load those 7 liquids into 7 of these 8 wells. What's important is you load 20 microliters and you follow all the tips and tricks of loading gels. If you break into one well, there's a chance you'll have a little bit of extra sample, and you can make yourself a note and use that last empty well to run that sample again so that you have a backup just in case you did break through the bottom. You've got that one backup well. That's really all you need to know. Follow the directions. This is pretty straightforward. Make sure to be careful of your surroundings because shaking that gel box, bumping into it, puncturing the gel, that's what's going to really make or break you in this part of the lab.